The last half of this course is really going to focus on the metabolism of carbohydrates and fats in order to generate energy for the synthesis of other molecules. And you'll see that it all comes down to protein enzymes that help mediate this process. We're going to look at metabolic pathways in the cell, and they can either be described as anabolic or catabolic pathways. And it all comes down to their dependency on energy, whether they're requiring energy or producing energy. We'll just look at, in general, how enzyme-catalyzed reactions are organized within the various pathways, but whether or not a reaction proceeds in the forward or reverse direction really comes down to thermodynamics, which we'll discuss further in the next lecture. If you recall, enzymes simply speed up the rate of a reaction and don't affect the thermodynamics of a chemical reaction. We've seen a few ways that proteins and enzymes can be modified, and this can regulate their activity. And when you regulate a particular step in a pathway, this is going to have global effects on the network of metabolic pathways. And lastly, we'll look at this idea of phosphoryl transfer potential, which is really why ATP has so much energy. These are the suggested readings for this introductory lecture on metabolism. What I hope is that after you've completed this course, you can really appreciate why biochemistry is essential for cellular function. I'm sure everyone's binge watching a lot of Netflix or things online, but if you're actually watching TV, you'll see commercials for multivitamins. You're going to hear a lot of claims in the media and the world around you, and your job as an educated university student is to think critically about the claims that are being made. These vitamins claim to be a complete and new way to support your energy, immunity, and metabolism, but then state that these statements have not been evaluated by the Food and Drug Administration. But there are, in fact, B vitamins that can act as cofactors for enzymes that will help with the metabolism of fat, carbohydrates, and proteins, and we've already looked at the role of cofactors in enzyme metabolism. So part of what they're saying is true, and this image of metabolism as a cycle is also correct. We are constantly making and breaking down molecules, and this really is the definition of metabolism, not just being able to break down foods. For instance, if someone says, I have a high metabolism, meaning I can eat anything. Enzymes are these specialized proteins that play a vital role in all of these processes. They are biological catalysts that can speed up the reactions a million times faster, allowing us to be able to break down the complex macromolecules like carbohydrates, lipids, and proteins into the waste products, carbon dioxide, water, and ammonia. And as we progress through the metabolism section, you'll see where the carbon dioxide and water comes from, and I hope you can appreciate where the nitrogen-containing ammonia waste product comes from as well. When you break the chemical bonds in these macromolecules, we're going from larger ordered structures to smaller ones, so more disorder in the system, and energy is going to be released. Instead of losing the energy as heat, we can store it in the bonds in ATP, and then we can use it for processes in the cell and body that require ATP. For instance, muscle contraction or the synthesis of larger molecules that a cell needs. There's also active ion transport, which we saw in the transporter lecture, and processes like detoxification, so to get rid of maybe drugs or other compounds that can harm the cell, as well as the production of heat and thermogenesis will help maintain body temperature and prevent the denaturation of proteins that are also energy dependent. So whenever you use an ATP molecule or adenosine triphosphate, you're going to hydrolyze a bond and produce ADP, or adenosine diphosphate, and an inorganic phosphate. We can then regenerate the ATP in the metabolic pathways, and we'll eventually see why oxygen is so very important for producing ATP in the mitochondria. What we need to do when we eat food is harness energy and channel it into biological work. Within a cell, it's the catabolic pathways, shown on the left, that break down all the nutrients we take in, so carbohydrates, fats, and proteins, and stores the energy found in their chemical bonds as ATP, as well as another molecule called NADPH. NADH and FADH2 are also two other high-energy intermediates that we'll see that can be used to make ATP. Now, these catabolic processes are oxidative in nature, producing the waste products that we excrete, and they're also exergonic, meaning they release energy. So we're degrading larger molecules to more simpler compounds, and instead of losing energy as heat, we're going to store it in the bonds found in these high energy intermediates. Anabolic pathways, on the flip side, are going to use that energy from catabolism and produce more complex molecules that the cell needs, or even store some of the energy in energy-rich compounds, for instance, making triacylglycerides from fat storage. And these processes are going to be using simple precursors like amino acids, sugars, fatty acids, and bases in chemical reactions that require ATP or NADPH, meaning that they're endergonic or energy requiring. These biosynthetic processes also tend to be reductive in nature. Bioenergetics really is the flow of energy in metabolism, where we're breaking down compounds and releasing free energy, but instead of losing it as heat, we're going to store it in the bonds found in ATP. We can then use that energy in order to synthesize larger structures that the cell needs. Organisms can differ widely in their source of energy and the biosynthetic precursors that they're going to use. We can classify organisms based on how they obtain energy, so either from the sun as light energy in the case of phototrophs, or from the oxidation of chemical fuels for chemotrophs. So these could either be organic or inorganic molecules. We can then further divide organisms into how they get their carbon. So if they can use carbon dioxide, they would be autotrophs. And if they require some other preformed organic nutrients, then they would be heterotrophs. Plants would be an example of a photoautotroph because they're using sunlight and carbon dioxide, while we humans would be chemoheterotrophs, feeding off of the photoautotrophs for our energy as well as our source of carbon. Metabolism really encompasses all of the chemical transformations typically carried out by enzymes to allow organisms to grow, reproduce, and respond to the environment. There are hundreds of discrete pathways, and this is just representing the intermediate metabolites or molecules as dots joined by enzymes that convert them from one molecule to another. You can see that some of the pathways are linear, where a starting molecule gets converted in a series of steps, and you might also see a branch point where another reaction takes an intermediate into a different pathway. Some of the pathways are also cycles, meaning a molecule comes in, goes through a series of reactions, and the final product of the pathway will then react with the starting substrate. You can get versions of these metabolism maps online, and they even used to sell them at those poster sales on campus. Now, back when I was an undergrad, one of my friends had it in his room, and he wanted to memorize every single step. So this really shouldn't be your goal because you could just look them up online, either through Sigma Aldrich or Roche using this link. But what is important is that you understand how the pathways are integrated. So if we represent all of the metabolic pathways in a cell as a map, we can see that it is quite complex, even more complex than some of the signaling pathways. And that there are some pathways that are linear, while others might be cyclic or branch off into the breakdown or production of other molecules. Some enzymes may only catalyze one reaction, while others might catalyze a series of reactions. And I'm just showing you an example of this, the pyruvate dehydrogenase complex, or PDC for short, which we've mentioned before in tutorial. It actually requires several cofactors and can catalyze 3 reactions in order to oxidize pyruvate to acetyl-CoA. And it's also going to produce that reduced coenzyme NADH. At the top is the cryo-EM structure of bovine kidney PDC, and because it is a complex of enzymes, it contains many copies of the E1, E2, and E3 subunits that catalyze these different reactions. What's really neat about this complex is because you are keeping all of these reactions in a very close proximity area, we're essentially channeling the intermediates from one active site to another. We will revisit this enzyme complex in a later lecture, so you don't need to know all these details. Just appreciate how complex enzymes can be. Enzymes can also associate into super macromolecular complexes, forming what's called a metabolon. Okay, so this would be a bunch of different enzymes that can associate with one another. And some of the citric acid cycle enzymes have been found to associate with one another. And again, this is going to facilitate the transfer of the product of one reaction to another. So in order for a cell to maintain homeostasis, it's really important that the pathways are regulated so that a cell can really respond to its needs. Let's look at a simplified linear pathway where we start with substrate A, and it's going to be converted to B in a chemical reaction, eventually producing a product F in the pathway. We're going to be representing the protein enzymes by E1, E2, E3, E4, and E5, and these are going to be catalyzing those reactions. We could also refer to A, B, C, D, E, and F as metabolites or molecules that are going to be produced and present in the cell. Now, there are a couple of ways we can regulate the pathway. So the most obvious one would be the availability of substrate. So if you didn't have any of A, then obviously we couldn't make F as the final product. We also need the enzymes to help catalyze or speed up these reactions. So their expression, folding in whether or not there are any mutations, or the presence of any required cofactors is also going to be important for the reactions to occur. We could have feedback inhibition where the final product F can inhibit an earlier step, or we could have molecules that can allosterically inhibit or stimulate the enzymes by binding and changing their structure. Cyclic AMP binding to protein kinase A, which we saw in the signaling pathway, was a way of changing protein kinase A's structure in order to release and activate the catalytic domains. We can also see modifications, such as phosphorylation, that can also inhibit or stimulate an enzyme. Now, the last way you can regulate an enzyme is through its degradation. So say E2 is degraded, then that might mean B starts to build up, and this could either slow down E1, or B could go into another pathway in order to produce X. So metabolism really is super complex because if you block one step, this can prevent the formation of an important product and lead to the buildup of metabolites or other toxic molecules. So this is just my cartoon summary of the main pathways that we'll briefly cover in this course. And what's really important for you to understand is the integration of these pathways. You can follow the arrows that represent enzymes and look at the breakdown of either carbohydrates, lipids over here, and proteins, and follow the paths all the way to the production of energy down here in oxidative phosphorylation. Some pathways also require energy, and these would be considered anabolic pathways, such as GNG, which stands for gluconeogenesis, and we'll look at it later on in this course and see how it makes glucose, as well as the synthesis of fatty acid chains for the storage of fat, and this will involve a central molecule called acetyl-CoA. So you can see that acetyl-CoA comes from glucose, so this is the link where eating too many carbs can make you fat. And acetyl-CoA is a great example of what's called an amphibolic intermediate, where amphibolic just means it can be found in and join both anabolic and catabolic pathways. Now, even though GNG and glycolysis are represented as separate pathways, they actually share some of the same chemical reactions. So we do need to review thermodynamics because this is going to determine if a reaction goes in the forward or the reverse direction. When we talk about the metabolic flux, it's the conversion of molecules or metabolites through a pathway, whether it be a linear or cyclic pathway, and enzymes can help speed up these reactions. They can also catalyze reactions that normally wouldn't be spontaneous through coupling to ATP hydrolysis. Thermodynamics drives these chemical reactions where a negative free energy change will indicate that a reaction will proceed in the forward direction. And you can also apply this logic to complete pathways. You can look at all the free energy changes for individual reactions and see if they add up to an overall release in free energy to make a final product. In cells, it's the standard free energy change that in the concentration of metabolites that will influence whether or not a reaction will occur. And the enzymes can also be regulated to prevent it from happening. This can dictate whether or not a pathway is on or off, or the metabolic flux, and we'll keep coming back to the importance of regulation and how enzymes can be regulated in the metabolism section. When we look at the hydrolysis of ATP and the amount of energy that's released, this is called the ATP phosphoryl transfer potential. We represent the change in free energy as delta G naught prime in biochemistry. So this is the Gibbs free energy change, and for ATP hydrolysis, it's a negative value, meaning energy is released when the phosphate is transferred off of ATP, in this case into water. The phosphate could also be transferred onto another molecule, but we would see a different amount of free energy released depending on the structures that are present. Now these are the standard values for the hydrolysis in water. So if you mixed 1 molar concentrations of all substrates and products, you would see that all of these reactions do proceed in the forward direction and energy is released. So that's why I've just written them as forward arrows. When you hydrolyze the different phosphate bonds, you actually get different amounts of energy released. And this is due to the stabilization of the products. It's the repulsion in the substrates that make them unstable and want to break apart, releasing free energy. So just coming back to one of our major themes in this course, it's the structure of a molecule that's very important for its function. In this case, the ability to carry energy. This change in free energy is what can be used to drive unfavorable reactions, such as those requiring energy in anabolic pathways in order to synthesize larger, more stable molecules. We're going to see these reactions come up over and over again in the metabolism section, so do make sure you know where these numbers are coming from. This slide is showing you the structures of the molecules, and remember, the amount of energy that is released is dependent on the differences in stability between the substrates and products. When we look at ATP, you can see that adenine base, the ribose sugar, and then 3 phosphates linked either in a phosphoester bond or 2 phosphoanhydride bonds. There's a lot of repulsion between the negatively charged phosphates, and when you hydrolyze and release this gamma phosphate, there's actually resonance in the free inorganic phosphate to stabilize it. But there's still a little bit of repulsion in that ADP product. So 31 kilojoules per mole of energy is released as these 2 products are more stable than the starting substrate ATP. When ADP is hydrolyzed, we actually get even more energy released since both of the products have resonance stabilization in their phosphates. It's not a big difference, but you might wonder why we don't see this reaction happening since more energy is released. But what it comes down to is how ATP is made in the mitochondria. Only ADP can be used to make ATP. If you were to start with AMP, which would be the product of ADP hydrolysis, you actually have to sacrifice another ATP molecule to donate a phosphate onto AMP, producing ADP for use in the mitochondria. This slide shows you the resonance forms of inorganic phosphate, which is also known as orthophosphate or PI. And if you remember the pKa values of phosphate, you can rationalize that this structure with 1 hydrogen on it will be the predominant form at physiological pH. And this allows the double bond to move between the various oxygens. So this is what creates resonance or stabilization in this product relative to the substrates that had repulsion between the negative charges. So if we just come back to these Gibbs free energy changes, ATP to ADP releases 31 kilojoules per mole of energy, while ADP to AMP releases a bit more, since both AMP and PI are more stable relative to ADP. AMP is already a pretty stable molecule, so when you remove that last phosphate, we only get 14 kilojoules per mole of energy. The last reaction is the hydrolysis of pyrophosphate, so just 2 phosphates linked together. And it can also generate a huge amount of energy, and this is going to be important for some of the pathways, which we'll see later on. So we see that the hydrolysis of the different phosphoanhydride bonds, or the phosphoester bond, can generate different amounts of energy, and it's all due to the difference in stabilization between the substrates and products. We can then use this energy to activate other molecules and drive reactions that are unfavorable or found in anabolic pathways that require energy. So we have this very important nucleotide that is made up of an adenine base, a ribose sugar, and three phosphates that are linked in two different kinds of bonds: phosphoanhydride bonds for the two terminal phosphates, gamma and beta, and a phosphoester bond linking the alpha phosphate to the sugar ring. ATP is involved in both catabolic and anabolic pathways through these phosphoryl transfer reactions, storing energy in those phosphate bonds due to the instability of the structure. Keep in mind that the amount of free energy released can also change since phosphates are ionizable. So if you were to change the pH, you can stabilize that repulsion. And metal ions can also stabilize the repulsion and affect the amount of free energy that is released. One last thing I want to mention is that nucleotides have similar phosphoryl transfer potentials. You can pass phosphates back and forth between the nucleotides and not lose any energy. So this is how other nucleotides get made. As the level of ATP rises, they can pass their phosphates onto GDP to make GTP, and you can also see GTP being used as energy molecules in certain reactions. When GTP is hydrolyzed, it releases the same amount of energy, meaning its standard enthalpy of hydrolysis is minus 31 kilojoules per mole. The enzyme that helps speed up this reaction is called nucleoside diphosphokinase, where nucleoside refers to the base in sugar, and diphospho means molecules with two phosphates on it, and then the kinase activity is just the ability to transfer phosphates between molecules. If you look at the thermodynamics of this reaction, you'll see that under standard conditions, it is reversible, so it's all dependent on the concentration of molecules, which will push the reaction in the different directions to balance out the concentration of nucleotides in a cell. So metabolism ultimately consists of both catabolic and anabolic pathways. The flux through the pathways can be regulated at the level of enzymes, allowing them to be turned off or on in order to switch between the anabolic and catabolic pathways, producing other metabolites that the cell needs. A great example of this would be diverting that amphibolic intermediate, acetyl-CoA, from the breakdown of glucose in order to produce or synthesize fats. Bioenergetics really is the flow of energy in the cell, where we're storing it in ATP and to a lesser extent some of the other nucleotides, but then the cells can use these in order to make larger macromolecules as it grows and divides. Make sure you understand how ATP stores energy before attempting the BBQs on Quercus.